Hello everyone, I am Nivedita Jena from Ambika Prasad Research Foundation. Welcome to all of you to our channel Biodiversity and Conservation. So today we are going to discuss about the Terminalia chebula, its habitat, its nomenclature, and about its pharmacological action and medicinal uses. So let's start. The Terminalia chebula, commonly known as black or chebulic myrovalan in English and Harida in Odia, is a species of Terminalia native to South Asia from Pakistan, India, and Nepal east to southeast China, and south to Sri Lanka, Malaysia, and Vietnam. And this species is held in high esteem in Ayurveda for its properties to prevent and cure diseases, and it has enjoyed the prime place among medicinal herbs in India since ancient times. Uh, Terminale chebula is a medium-sized deciduous tree with a height up to 30 meter, wide-spreading branches, and a broad rounded crown. It grows in the altitude of 1500 to 2000 meter in mostly clayey as well as sandy soils. Here is the nomenclature of Terminalia chebula, in which kingdom is Plantae. The division is Magnoliophyta, class Magnoliopsida, the order Myrtles, family Cumbrithaceae, the genus Terminalia, and the species is Terminalia chebula. Terminalia jebula is found mostly in mixed dry deciduous forest and is frequent in tropical and subtropical zones, mostly in hilly tracts. The plant prefers tropical environment, ascending in the sub-Himalayas zones up to an elevation of 1500 meter. And the habitat includes, in the natural habitat of the species, temperature ranges between 36 degrees centigrade and 45 degrees centigrade, and rainfall ranges from 1200 mm to 3000 mm per annum. And it is capable of growing on different types of soils but attains best development on loose, well drained soils such as sandy loam as well as clay loam, and it grows in open areas in forest, forming top to middle canopy. Then coming to its morphological characteristics, Terminalia chebula is a tree species, tall about 15 to 24 meter in height, and it has round crown and spreading branches. Then the leaves are usually subopposite, ovate or elliptical, with silky hairy when young, and in mature stage, the petiole is about 1.2 to 2.5 cm usually, with two glands near the top. Then the flowers are monoecious, dull white to yellow, very densely white billows within and having a strong unpleasant odor. Then the fruits are about 1 to 2 inches in size, has 5 lines or 5 bricks on the outer skin. They are green when unripe and yellowish gray when ripe. And the bark is gray or dark gray with some longitudinal cracks, particularly in the old trees. So basically, the flowers and fruits appear in March to April and November to December respectively in Terminalia chebula. Then the ethnomedicinal uses of Terminalia chebula are the following. The fruit is mild laxative, stomachic, tonic, alternative, antispasmodic, and it is useful in ophthalmia, hemorrhoids, dental caries, bleeding gums, ulcered oral cavity. And the fruit paste with water is found to be anti-inflammatory, analgesic, and having purifying and healing capacity for wounds. And its decoction is used as gargle in oral ulcers, sore throat. And the fruit powder is a good astringent dentifrice in loose gums, bleeding and ulceration in gums. It is good to increase appetite, digestive aid, liver stimulant, stomachic, gastrointestinal prokinetic agent, and mild laxative. And the powder of Terminalia chebula fruits has been used in chronic diarrhea. It is used in nervous weakness, nervous irritability. It promotes the receiving power of five senses, and it is adjuvant in hemorrhages due to its astringent nature and good for chronic cough, sore throat, as well as asthma. Also, it is useful in renal calculi, dysuria, retention of urine, and skin disorders with discharges like allergies, urticaria, and other erythematous disorders, etc. Then coming into its pharmacological actions. So the first one is antioxidant activity. The leaves, bark, and fruit of Terminalia jebula possessed high antioxidant activity, and phenolics were found to be responsible for this activity. And Terminalia chebula in a polyhobal formulation inhibited free radical induced hemolysis and also significantly inhibited nitric oxide release from lipopolysaccharide, stimulated murine macrophages. Six extracts and four compounds of Terminalia chebula fruit exhibited antioxidant activity at different magnitudes of potency. Strong antioxidant activity of aqueous extract of Terminalia jejula was observed by studying the inhibition of radiation-induced lipid peroxidation in rat liver microsomes at different doses, and methanolic extract was also found to inhibit lipid peroxide formation and to scavenge hydroxyl and superoxide radicals in vitro. And then the anti-carcinogenic activity. A group of researchers have reported the inhibitory action on cancer cell growth by the phenolics of Terminalia jejula fruit and found that gibberellic acid, tannic acid, and ellagic acid were the most growth inhibitory phenolics of Terminalia jejula. Ethanol extract of Terminalia chebula fruit inhibited cell proliferation and induced cell death in a dose-dependent manner in several malignant cell lines, including human and mouse breast cancer cell line and human osteosarcoma cell line, human prostate cancer cell, and a non-tumorigenic immortalized human prostate cell line. Beside acetone extract of bark and fruit powder of Terminalia jejula harbors constituents with promising anti-carcinogenic activity. Then hepatoprotective activity, a mixture of gibberellic acid and its minor isomer, nucleophilic acid, with the ratio of 2:1, isolated from ethanolic extract of Terminalia jejula fruits, showed strong hepatoprotective activity. And ethanol extract of Terminalia jejula was found to prevent the hepatotoxicity caused by the administration of rifampicin, isoniazid, and pyrazinamide in combination in subchronic model. And protective effects of an aqueous extract of Terminalia jejula fruit on the tert-butyl hydroperoxide induced oxidative injury was observed in cultured rat primary hepatocytes, and rat liver have also been documented. Terminalia jejula in a herbal formulation showed hepatoprotective activity against carbon tetrachloride induced toxicity in rat hepatocytes. And then the cardioprotective activity. The Terminalia chebula extract pre-treatment was found to ameliorate the effect of isoproterenol on lipid peroxide formation and retained The activities of the diagnostic marker enzymes in isoproterenol induced myocardial damage in rats. Its pericap has also been reported to have cardioprotective activity in isolated frog head model. Terminalia chebula exhibited antibacterial activity against a number of both gram-positive and gram-negative human pathogenic bacteria, and ethanoic acid and ellagic acid as isolated from butanol fraction of Terminalia chebula root extract had a strong antibacterial activity against intestinal bacteria, Clostridium perfringens and Escherichia coli. It is effective in inhibiting the on-urease activity of Heliobacter pyrrolium ubiquitous bacterium implicated in the development of gastritis, ulcers, and stomach cancers. Ripe seeds of Terminalia chebula also exhibited strong antibacterial activity against S. aureus, which is Streptococcus aureus. The aqueous extract of Terminalia chebula strongly inhibited the growth of Streptococcus mutans, salivary bacteria. Diffused of Terminalia chebula showed an inhibitory effect against strain X100 of the bacterium Xanthomonas campestris, indicating its usefulness for the management of citrus cancer disease. It has also growth inhibitory action against Salmonella typhi, Klebsiella, Shigella, and intestinal bacteria. And ethanol extract of Terminalia chebula fruit showed strong antibacterial activity against multi-drug resistance, uropathogenic Escherichia coli, and phenolics were found to be responsible for this antibacterial activity. Then the antiviral activity. So the terminal chebula fruits afforded for 4 immunodeficiency virus type 1 integrase inhibitors, um, GA and 3 galloyl glucoses. Their galloyl moiety plays a major role for inhibition against the reprocessing of HIV-1 integrase of the compounds, and the Terminalia chebula has also retroviral reverse transcriptase inhibitory activity. It protects epithelial cells against influenza A virus, supporting its traditional use for aiding in recovery from acute respiratory infections. And the methanol and aqueous extract of Terminalia chebula showed a significant inhibitory activity on human immunodeficiency virus 1 reverse transcriptase. It also demonstrated the therapeutic activity against Herpes simplex virus both in vitro and in vivo test. This finding prompted a team of Japanese researchers to investigate Terminalia chebula's effect on cytomegalovirus. They found that Terminalia chebula was effective in inhibiting the replication of human cytomegalovirus in vitro and in an AIDS model with immunosuppressed mice, and concluded that it may be beneficial for the prevention of CMV diseases and immunocompromised patients. It is also helpful in sexually transmitted diseases and AIDS. And tannins from Terminalia chebula are effective against potato virus X. Thank you.